and I welcome all of you in this special public webinar, which we are organizing as part of the School Outreach Program. It's jointly organized by the National Academy of Sciences India Delhi chapter and the Indira Upadhyay College University of Delhi under the aegis of the DBT's Star College program. Today we have with us Dr. Subrata Kundu from School of Chemistry, Indian Institute of Science Education and Research, Thiruvananthapuram, and he'll be talking to us in next 30 to 40 minutes on a topic, small molecules and metals in life processes, and overview. So before inviting Dr. Subrata, let me briefly introduce him to all of you. He was born and raised in Barrackpore, West Bengal, India, and received his Bachelor of Science in Chemistry from Jadavapur University in 2007. And then he moved to IIT Kanpur for Master of Science in 2009. He completed his Ph.D. in Inorganic Chemistry and the highest grade, that is summa cum laude degree, from Germany. Subsequently, he accepted an offer to join a postdoctoral position in the group of Professor Tim Warren at Purdue and Georgetown University. Since July 2017, he is working as an assistant professor at IISER-Tvm. A research group led by him utilizes synthetically accessible bioinspired coordination complexes, aiming to provide insight into the molecular basis of nitrous oxide and hydrogen sulfide signaling processes in mammalian biology. With these words, I now request Dr. Subrata to kindly share his screen to us talk.
Yeah, thank you very much, Professor Manoj. And now I'll be sharing my screen, and then we'll start. Yeah. So now, good afternoon, everyone. Today, as Professor Manoj Saxena already introduced, that today we'll be talking about various small molecules, but we would be discussing their chemistry from biological point of view, and we will see that how metals affect their chemistry. And so mostly I will be giving you the overview, and some of the technical thing I would be trying to avoid, but definitely if any of you have further questions on the technical aspect or any other aspect, you can find me from our website and you can write me. I would be happy to answer those as well. So now, first I would like to start with one of the molecule which created a huge panic over the last couple of months. And this slide perhaps tell you the couple of screenshots taken from various newspapers, that what we have seen that various countries and various states were just like pushing different types of oxygen tankers from one place to other just to support the oxygen crisis, right, in different hospitals and then different other health facilities, right? So that's why first I would like to start with the molecule oxygen and how it binds to the metal and how it gets transported in our body. And then we'll be also seeing why we need oxygen for various metabolic purposes. Some of the, again, key examples. We really do not have time to go behind each and every one. But again, I thought of starting with oxygen because we, many of us who do not practice chemistry even got familiar with the term oxygen in recent weeks or months, right? So now, so these are the different oxygen carriers or what is present in various species. For example, you can see at the leftmost position. So this is for mammalian biology, including definitely human. You can see that hemoglobin is the molecule as highlighted over here, which carries basically oxygen from one part of the body to another part, right? So, and this is the molecule where you can see iron is present. and it is hosted inside a big molecule. Let us not worry about that at this particular slide. We'll go into details in the later slides. And this particular molecule, as we all know, that appears red, right? And this is not again red for all the animals, right? For example, you can see this color of blood can be blue for certain species. For example, you can take octopus, you can take squid, or even there are many other marine species which you can consider. Again, in their case, the blood, and by the word blood, I would like to mean that the oxygen carriers. In those species, the oxygen carrier would look blue, and then this shade of the blue is typically the color which is indicative of their blood color. And you can see in this particular case, you do not have the structure of the hemoglobin, which is present in case of mammalian system, or in other words, in case of human beings, right? So rather, in case of this kind of species like octopus, you have hemocyanin. The name of this molecule is hemocyanin. And then this kind of metalloproteins are involving copper sites in it instead of iron, unlike the mammalian system. And in this case, you can see the oxygen is bound in between two copper sites, and then it appears blue. And now, likewise, there are a couple of other marine animals or worms. You can see in some cases the blood, or in other words, oxygen carrier may appear green. And then there are animals where it would appear violet. And again, depending on the species or depending on the oxygen carrier, they would be utilizing various metalloenzymes, and they involve different kinds of metal. For example, again, hemoglobin involves iron, then hemocyanin involves copper. Then likewise, there are other iron-containing enzymes, but definitely not same as hemoglobin. They also can carry oxygen in case of different species. Now, in the next slide, what we will look into details is hemoglobin. And again, what we will be seeing is this particular molecule with a closer look over here. And then over the time, what I would like to emphasize is that how a chemist can look into this kind of molecule, and then a synthetic chemist more specifically can make certain models of this and investigate this kind of molecule from like the chemistry laboratory point of view to understand some of the biological aspect. So now, before we go further, let us emphasize couple of keywords over here. So as the name suggests, that hemoglobin and myoglobin, so both are so one of them is oxygen carrier, another is like oxygen storage protein. And again, in both the name, you can see that the common part is globin. And what does this globin mean? Globin is nothing but a peptide chain which contains 135 amino acid in it. Now, in case of myoglobin, which would be abbreviated as MB, you will be seeing that this particular metalloprotein is structurally very similar. I would like to mean with that is that this has similar active site, and by the word active site, I would like to mean the place or the site where the oxygen binds. Okay, and in this particular case, what you see is that in case of myoglobin, this is oxygen binding protein, and this is found in muscle tissues. So that means again in muscle tissues, the oxygen concentration would be less, right? So this is the place where myoglobin can be found, and again this is for the storage purposes, right? And then hemoglobin is the metalloprotein which transports oxygen. Again, it will take oxygen from lungs, and then it delivers to the myoglobin. And again, myoglobin is located at the various tissues, and where the oxygen would be stored, right? And then structurally, as I said, the active site of both hemoglobin and myoglobin in next couple of slides, so we'll be seeing that closely. But in short, what you would see is that the active site would contain in both the cases this particular moiety. And then in one case, it is tetrameric. For example, you see over here as written that hemoglobin is a tetrameric unit. So that means 4 of such kind of moiety would be present, definitely in presence of the globin part. And then in case of myoglobin, only one unit would be there. As a result, you see that there are differences in terms of their molecular weight. One is 16,000 kilodalton, and another is almost 4 times 65,000 kilodalton. Okay, and then what you see at the bottom of this slide is the Nobel Prize awarding chemistry regarding this hemoglobin and myoglobin came out in 1962, and work was done by Professor Kendrew and Peruz, right? And then they identified the extracrystal structures of myoglobin and hemoglobin, and which later found to be very important to understand the chemistry of hemoglobin and myoglobin, chemistry and biochemistry of it. So now we are slowly moving closer to the protein or active site of the metalloprotein. And what you see over here is the truncated version of the active site, right? So again, in the previous slide, what you have seen is the active site. And now I am truncating all the substituents over here at the outer periphery. And rather I am focusing on the middle part of that ring, okay? So now if you look at this particular slide, the middle part of the ring contains this particular ligand, and I hope some of you at least would be familiar with the term ligand. So, and again, with the term ligand, we would like to mean that some certain chemical species which can bind to the metal would be termed as ligand. And then now this is the ligand, which is called porphyrin. This is a macrocyclic ligand which has 4 nitrogen donors, and it can donate, in this case of hemoglobin or myoglobin, it can be coordinated to the iron site. And then this is the central unit which you have in case of hemoglobin or myoglobin, right? So now definitely you have a lot of different substituents at the outer periphery. For example, these positions would be substituted with different other functional groups, and then accordingly they would be anchored to the amino acid chain, right? So now again, if you go a little bit bigger out of this initial coordination site of the iron, so what you see is this particular structure which is shown at the rightmost position. So now again, in this particular case, at the central position, you have iron, which is coordinated to 4 nitrogens of the porphyrin ligand. And then from the bottom of this particular plane, what you can see, one of the histidine moiety, which is nothing but another amino acid, which comes from the bottom. And this position is called as axial position because this comes through this axis. And then this plane of porphyrin would be called as equatorial position. So now this histidine comes from the axial position. And now, as you may know, that most of the transition metals, especially iron, cobalt, so this kind of transition metal typically prefers 6-coordinate geometry. Now, in this particular case, you can see that 5 of the coordination sites are getting fulfilled by 5 of the nitrogen. 4 nitrogens are getting satisfied from the porphyrin ligand, and then 1 nitrogen is coming from the histidine ligand from the bottom. So that means one more site is still vacant, and then this is indicated with this black square-shaped image, right? And this is the site where the oxygen binds in case of hemoglobin and myoglobin, and then depending on whether it is hemoglobin or myoglobin, their binding affinity will be different, and then they carry or they store the oxygen moiety. Okay, and again, perhaps I do not have time to elaborate completely that why carbon monoxide and cyanide binds also hemoglobin, and in fact they bind hemoglobin more tightly as compared to oxygen, and that is why we hear that cyanide poisoning or carbon monoxide poisoning, which leads to the deadly event whenever, for example, if someone has cyanide poisoning, or even if someone is having barbecue in a closed place or having some firework within a closed place, so they can have carbon monoxide poisoning because of very tight binding of carbon monoxide to the hemoglobin. And again, it binds very tightly to the iron site of the hemoglobin, and the primary reason for this is that the type of ligand, if you try to elaborate that what type of ligand carbon monoxide is, what type of ligand cyanide is, and then if you compare that with oxygen, so one can get the answer why they have higher toxicity or higher binding affinity. As a result, they do not allow further oxygen molecule to come in and then get transported to the other part. So So now this particular slide, what you see over here is a little bit colorful view of deoxy-myoglobin. And again, by that word deoxy would like to mean that when the oxygen is not bound to that myoglobin. And again, as I said, that the active site of this particular enzyme of this particular metalloprotein would have this iron porphyrin moiety, which is shown at the central position over here. And then from the axial position, you can see one of the histidine is coming. And now this entire moiety is basically hosted by these colorful ribbons, and these ribbons are nothing but the amino acid protein sequences, right? Amino acid sequences, and then through those protein sequences, different functional groups will be anchoring this porphyrin as well as this histidine from the bottom. And also you can see there is another histidine moiety which is hanging from the top. That also has some role, although that is not in the close proximity, or that is not in the distance which can directly bind to the iron site, but still it has some role over the time. We will be trying to see that how it influences, right? So now if we take a even closer look, so this is what we have seen again in the last slide, that you have the iron moiety which is embedded inside the porphyrin pocket, and then from the bottom you have one of the histidine which is in the close proximity of the iron. So as a result, this is called proximal histidine, and you can also measure the bond distance from the crystal structure of this protein crystal, and then you can see there is another histidine which is a little bit far, and that's why it is called distal histidine. And as a result, this is not doing any direct bonding with iron. And then one can also look into the details of the oxidation state, spin state. Perhaps we will not be able to discuss more in details about that. But again, this spin state and then oxidation states also play a very crucial role in terms of oxygen binding and transporting and so on. So now in case of hemoglobin, as I said, hemoglobin is nothing but the tetramer of this myoglobin. So as a result, whatever you have seen in this particular case, that you have the colorful ribbon, and then along with that you have one porphyrin moiety with couple of histidines in the close proximity. So now in case of hemoglobin, you see you have exactly 4 of such kind of units. So you see one ribbon is shown with the red color, then another ribbon is shown with this fluorescent green, another ribbon is shown with this cyan color, another ribbon is shown with the blue color, and then each of these different color indicates the monomeric unit which is equivalent to myoglobin. And again, you can see in each of the color of this ribbon, you see one such porphyrin moiety is basically hanging over there. And then again, this is the overall active site structure what you see in case of hemoglobin unit. Okay, so that is the basic difference in case of hemoglobin versus myoglobin. And as a result, so there are it's not only the structural difference, monomer versus tetramer. Of course, there are certain differences in terms of their activity, in terms of their oxygen affinities, and so on. If time permits, we'll try to look at that later on. So now, whenever we look at previously, you are looking at deoxy-myoglobin. Now, if we look at the oxy-myoglobin, so in case of oxy-myoglobin, what happens? So you have the again deoxy-myoglobin. You are looking at the porphyrin ring from one of the side. As a result, it appears like a planar ring. But you can see that iron is basically hanging out of the plane, right? So that means this is called out of plane geometry of iron. Now, once it binds oxygen, so that means O2 molecule. So what you see is that this iron is right now placed within the plane, within the porphyrin plane. Whenever you are looking at from the side view, you see that this iron, this brown shaded ball is exactly fitting in the pocket. So this is called in-plane geometry. And again, this is quite interesting from the structural point of view. And then if from chemistry point of view, if you want to know that how this happens, so in this case, one should look at the oxidation state. In this particular case of deoxy-myoglobin, you have iron in +2 oxidation state. And then in case of oxy-myoglobin, you have iron in +3 oxidation state. So now what you need to do is that you need to look at their size. So you need to think about what happens if you oxidize a metal site. So you will be seeing that the size of the metal decreases, because the nuclear charge which remains unchanged, but the number of electrons will get lower whenever you oxidize the metal site. As a result, the overall size of the metal ion gets lower. So as a result, in this particular case of oxidized molecules, so iron 3 fits better in the pocket of the porphyrin, right? So that's why you get the planar geometry. On the other hand, whenever it is in iron class 2 oxidation state, this is out of plane geometry. And again, this has significant consequence in case of their activity, because again, in short, what happens whenever the iron site is resting or iron site is held outside the plane of the porphyrin, this is called tense state. So that means the molecule itself is tensed. On the other hand, whenever the iron is held inside the plane, so that is called that relaxed state, right? So that means that has something to do or that gets translated into their activities and so on, right? Again, perhaps we do not have time today, but again, I recommend if you are interested, please go through a couple of textbook or different web-based material. So now on this particular side, on the right, what you see is the overall structure. And what you see is that iron is nicely sitting inside the porphyrin pocket. Then from the bottom, there is a histidine moiety, which is called as axial histidine or proximal histidine. And then, as I said in the earlier slide, that the histidine from the top, although previously it was not doing any job, right now you can see this started doing something. And what you see over here, this histidine has one of the NH moiety over here. I think in next slide or next slide, I have some scheme. You will realize that better. But what you see, there would be one NH moiety, and that does hydrogen bonding. So that means, again, there would be some weak interaction over here. But although this interaction is quite weak, but still this has some significant influence in terms of the overall structure, as well as the overall binding of the oxygen molecule. And again, from chemistry point of view or inorganic chemistry point of view, one can look at what is the oxidation state, and then what is the redox state of molecular oxygen, and so on. And then these are the different spectroscopic parameter one can get for this oxy-myoglobin. And again, more or less same holds true for the oxy-myoglobin as well. Okay. So now in the next slide, what you see, as I was trying to demonstrate, that what is the role of the histidine moiety, which is hanging on the top. But initially, in case of deoxy-myoglobin or hemoglobin, it was not interacting at all with the iron because it was too far from iron site. So what you see over here, this is one of the, these are two different variants of hemoglobin, right? Again, this particular one is the schematic diagram showing the hemoglobin from human blood. And then on the right hand side, you see this is the hemoglobin from this particular kind of worm, which can be found in soil and other places also. This is called nematode. This is the image of this particular species. And what you see over here is, in terms of activity, you look at this particular interesting data that the relative oxygen affinity for human hemoglobin is, if it is 1, then in that particular scale, you can see the relative oxygen affinity for the hemoglobin which is present in this worms, which is called nematode. You can see this is 10 to the 4 times, so which is significantly higher, 4 orders of magnitude higher. And what you can see is that the geometry at the iron site over here, what I am indicating with this red circle, is almost same. Okay, there is almost no difference. So that means the primary coordination sphere of iron, so that means the immediate neighbors of iron is exactly same in both the cases. But what you see is that this particular moiety over here, so in this case you have histidine, which was hanging on the top previously, and now in presence of oxygen, this is doing hydrogen bonding interaction as shown with these dotted lines. And in case of this nematode, what you can see is that instead of this histidine, you would have this tyrosine moiety, one equivalent of tyrosine as amino acid, and then another equivalent of glycine would be there. And now taken together, what you see is that the hydrogen bonding network, which is involved in this case of in this particular kind of molecules, would be totally different. So here you can see more number of hydrogen bondings, and then as a result, so the oxygen binding would be much more facile And that is what exactly you can see, that oxygen binding affinity of this kind of species gets significantly different. And again, this is something that biology has, or nature has designed it very precisely, because again, this kind of worm, they live in soil and so on, so where the oxygen concentration is quite less. So that means the affinity of this particular kind of oxygen transporter needs to be higher, then only they can bind oxygen and so on. On the other hand, in our case, especially whenever we think about the non-COVID era, we can think we can breathe in happily, and then we don't need to worry too much about, but of course nature has brilliant design behind it, right? So, but again, you can see, just based on the amino acid residue which is placed outside of this this primary coordination sphere of iron, again, by the word primary coordination sphere, I would like to mean the immediate neighbors of iron basically makes the primary coordination sphere, and anything beyond that is something which makes the outer coordination sphere. Okay, so now in the next slide, what you see is that what we can do from chemist point of view, especially as a synthetic chemist, what can be a role of synthetic chemist in this connection of hemoglobin, myoglobin, or even anything in connection of bioinorganic chemistry. So what one can do is that you can take this particular active site, which is the iron porphyrin moiety at the central position as highlighted, and that particular moiety one can make in the lab, and it is quite easy to make this molecule. So you can take this pyrrole, and then you can condense that pyrrole in presence of aldehyde, and one can get this kind of this is called tetraphenyl porphyrin. And definitely you can do the metallation with iron, and then you can get the iron compound, and then you can study the oxygen chemistry, and you try to understand that how this kind of model can be used. So again, this would be called as model, because you are not exactly making the same molecule as in biology, but you would have this kind of phenyl rings and other kind of substituents over here, just to understand or whatever is possible from synthetic point of view, and then you would be employing these particular models for understanding some of the biological activities. So now again, one can make this quite easily, and then this kind of reactions are nowadays quite easy, and it can be made successfully in a very pure form. But the problem is that whenever you try to study this chemistry of hemoglobin or myoglobin by using the model system, but again, what you need to think of is that this is a planar molecule. As a result, so I can represent this particular molecule with this particular kind of cartoon diagram, where I have a plane represented by this black solid lines, and then inside I have iron in class 2 oxidation state. So now what happens if someone makes this model in the lab, synthetic laboratory, and then you try to study the oxygen chemistry? So instead of getting what nature does, so you will be getting this particular moiety, right? So that means there will be a peroxide moiety, which is bridging two of the iron 3 sides, right? So that means two of the porphyrin would be just like parallel to each other, more or less, and then in between there will be a peroxide bridge, right? And then this particular peroxo compound over the time decomposes to give you this kind of oxo compound, and then eventually this is one of the useless compound. It does not have any implication in terms of this reversible binding of oxygen and so on. So that means whatever we can do in the synthetic laboratory is far worse as compared to what nature does, right? Probably it does not need any justification. We all know that. But again, this is another example. But again, we want to be better, right? So as a result, what one can do is that one can try to take lesson that what are the things we could do better, right? One of the thing is that, as I was trying to emphasize, that in previous case also you have seen that there is one histidine moiety from the top which comes and does hydrogen bonding, and I was trying to emphasize that has significant influence in terms of oxygen binding and so on, right? So now in this particular case, whenever we make this molecule in lab, we do not have such kind of histidine moiety which comes from the top, right? And then does the hydrogen bonding and so on. So now to overcome that problem, so what we can do is that we can tune this particular R group, or we can change this particular R group over here on this phenyl rings, and then we can try to make some hydrogen bond donors. So that means, again, it may not be exactly same as histidine, but again, it can be something which can donate hydrogen bond to support the hydrogen bonding to understand the natural system. So to do so, so this gentleman from Stanford University, Professor Coleman, he made this molecule right in his lab, and you can see instead of simple R group like methyl, he made this particular kind of amide kind of molecule or substituents, right? And what you can see that this particular NH moiety can do the hydrogen bonding. Again, this is exactly not same as the histidine as biology employs, but again, it is something better than the previous or primitive model where you did not have any hydrogen bond donor. In this case, actually, you have some hydrogen bond donor which is coming from this particular arms, right? Ligand arms. As a result, you can see there are hydrogen bonding as shown with the dotted lines. And actually, this particular molecule, whenever you have this hydrogen bonding network, it can actually bind hydrogen in a reversible fashion, right? Again, you look at the previous slide. In previous case, as I said, that it binds oxygen, no problem. But again, it does not bind it reversibly. Eventually, it goes to this iron 3 oxygen, iron 3, which is called iron 3 iron compound, which is again will not allow you to do bind oxygen in a reversible fashion. But again, in this case, just by changing the hydrogen bonding scenario in the outer coordination sphere, one can make the reversible binding of oxygen. So that again tells that not only the coordination site which is immediately next to the metal, it's not only that particular coordination site, even the outside of that, so that is which is called secondary coordination site is also important. Okay. So now, while we are discussing the binding of oxygen, so one should also think about definitely through lungs we get the oxygen, right? And then hemoglobin transports the oxygen to different tissues. Then myoglobin will be storing that oxygen after taking it or after snatching it from hemoglobin. And definitely the myoglobin will not hold that forever, right? So that particular oxygen will be getting utilized for the metabolism of food, then for the metabolism of other molecules in our body, right? So now the question is that how does that happen, right? So for understanding that, what you probably need to understand, and again, it might be beyond the current knowledge of some of you, but again, some of you, of course, will know it, is that the molecular orbital diagram, which is again very simplified one, right? So, so what you can take is that whenever you are considering the molecular orbital diagram of oxygen molecule, you can see you need to take the atomic orbitals of oxygen in the both sides, because again, this is a diatomic molecule, and then you need to use the linear combination of these atomic orbitals of oxygen, and then you can get the molecular orbitals of molecular oxygen, of course, and then you will be getting different bonding and anti-bonding energy levels. And what is most important for us is this particular levels over here, which are called anti-bonding orbital. Why these are most important for us for today's discussion? Again, the reason we are most interested about those particular orbitals, because we want to utilize the oxygen. Now, if you want to utilize the oxygen, what you need to do is that you need to activate oxygen, right? Now, the question is that how to activate molecular oxygen. Again, by the term activation, what we would like to mean is that you have the stable molecule. Now you want to destabilize the molecule just by breaking the bond, right? Breaking the oxygen-oxygen bond and so on. So now, at least whoever are at least familiar with the primary concept of molecular orbital diagram, what you know is that one can calculate what is the bond order of oxygen, molecular oxygen. Again, if you see molecular oxygen, you have bond order of 2, and you can get that very easily, that if you take the number of electron in the bonding molecular orbital minus number of electron in the antibonding molecular orbital divided by 2. That is how you can get the bond order of any given molecule. But again, in this case, you can get the number 2 very easily by using this particular formula. So now, to cut a long story short, what you need to do is that to utilize that molecular oxygen, you can add electron, and if you add electron to the molecular oxygen, you will be getting superoxide. If you add further electron, you get peroxide. Again, in this case of superoxide, you have bond order of 1.5. In case of peroxide, you have bond order of 1.0. And then in case of oxo, you have bond order of 0. So that means starting from the bond order of 2, what we are doing is that as soon as you are populating the antibonding orbital, we are basically breaking the oxygen-oxygen bond, and then we are basically activating the oxygen, and then we are getting different reactive species. And this is how the different reactive species gets generated. And this reactive species will be utilized for the oxidation of different metabolites, right? And as a result, so we'll be doing the metabolism. But again, this has another detrimental consequence. For example, in terms of iron, so this kind of peroxide can also lead to the generation of free radical, right? And this can lead to the oxidative damage of the living tissue and so on. So that's why there needs to be a balance between how much oxidizing equivalent needs to get generated and how much needs to be then quenched by using the antioxidant and so on. I think there are a couple of questions in the chat box. I will take that perhaps towards the end. Yeah. So now in the next slide, what you see is that again this is a little bit busy slide. I do not perhaps have the time to go into the definition of all of this. But with this slide, what I wanted to share is that by using this molecular oxygen, and then with the help of proton and electron, and again this proton equivalent and electron equivalent comes from, for example, electron comes from different electron transfer proteins in the biological system. And then in terms of oxygen, what the enzymes like cytochrome P450 can do is that this can oxygenate this CH bond and can give you alcohol, right? So that means starting from alkane, you can get alcohol, right? And again, from organic chemistry point of view, this is a quite interesting reaction. And then another reaction can be that you have this kind of olefin, and again in presence of oxygen and 2 equivalent of proton, 2 equivalent of electron, in presence of cytochrome P450, which is the metal enzyme, in this case iron containing metal enzyme, you can get the epoxide. But again, these are some of the reactions we can definitely do in the synthetic laboratory. But one need to wonder that how this happens in the body, because this is very important, especially when you would be, for example, if you are looking at a new pharmaceutical molecule, if you want to design a new molecule for pharmaceutical purposes, you really need to think about how it goes inside the body. So that means how that particular drug gets delivered at the site. And then let's say it does the desired job of killing the disease. And afterward, you need to think about how it gets excreted from the body. So for that, you need to metabolize that particular drug molecule. And for that, you need to chop that particular molecule. Most of the time, it would be organic molecules, right? So that means you need to oxidize those molecules. And then you need to make sure that it gets solubilized in the solvent of our body, which is nothing but water, right? And then it gets excreted from the body. Now, for those purposes, you really need to think about how it gets oxidized, right? And then again, that happens by this kind of enzyme. And one of the most relevant one in this particular consequence is, or in this particular topic, is the cytochrome P450. Okay, so let us take one example that how. So this is the active site. How does it look like, right? So there is again the porphyrin moiety. You have iron at the central position in the pocket of the porphyrin, and then from the bottom you have a cysteine moiety, which is coming. But again, you see the difference. So again, you have similar porphyrin pocket. You have iron there. Up to that, it is exactly same as hemoglobin or myoglobin. But whenever you had hemoglobin or myoglobin, you had histidine from the bottom of the porphyrin. But here you have cysteine, right? So as a result, it does completely different job. In previous case, we are binding oxygen, and then just like a reversible binding, we just basically carry oxygen from one place to other. So for example, lungs to tissue. But in this particular case, we activate oxygen. As a result, we can form different active intermediate, which can oxidize the CH bond. For example, this is nothing but calciferol. So is one of the variant of vitamin D3. And this can get oxidized in presence of cytochrome P450 and oxygen, as you can see over here. And this particular enzyme oxidizes this particular CH bond, which is present over here, that gets converted to OH, right? And there are a couple of other places. For example, this CH bond also gets converted to OH, and then likewise, so this gets converted. A different CH bond of a given substrate gets converted or gets oxidized or gets hydroxylated. So likewise, you can see this is a paper, this is a reference from Nature journal. And in this case, again, what you see over here, this is nothing but the red highlighted portion is nothing but the cytochrome P450 active site. And then on the top over here, you see a drug molecule, which is the name of this molecule is warfarin. And this is a blood thinner, so anticoagulant, which is used for making the blood thinner for certain disease, right? And again, this kind of drug molecule can get hosted by the active site pocket of cytochrome P450, and then it will get metabolized. So now, likewise, this is also another crystal structure where you can see that this bottom portion is the active site of cytochrome P450, and then on the top you have the camphor. Camphor is nothing but perhaps we all know about what is camphor. We use for many purposes even in our daily life, right? So in this case, so you can see this particular CH or this particular carbon is located towards the iron site, and then as a result, so this CH bond gets hydroxylated, and as a result, you can see this particular CH bond will get hydroxylated, not other CH bonds in the camphor. Again, that is where the pocket of that metal enzyme plays a very crucial role in terms of determining the selectivity of the CH bond hydroxylation. This is the overall scheme. I do not have time to go behind, and also I think it may be a little bit beyond the scope of this particular lecture. But before I end, I just want to show one or two slides that so far we are talking about oxygen, but another molecule which is very close to oxygen. If you look at the periodic table, that if you chop oxygen bond and you replace one of the oxygen with nitrogen, you get this particular molecule, which is called nitric oxide. And if you count total number of valence electron, definitely in case of oxygen, you had even number of valence electron, although oxygen is a paramagnetic molecule. But in case of nitric oxide, you of course will have one unpaired electron because total number of valence electron is odd number. So now this molecule is also very interesting molecule from biochemistry point of view. And as you can see is that this molecule participates in various biological activities, including neurotransmission, vasodilation, and then immune response of our body and so on. But then at the same time, if the concentration, if you look at, if the concentration is high, so this will lead to the cell death. And then as a result, nitric oxide is called as a deadly molecule, right? So that's why I termed this molecule as a tricky molecule. And I think before the 1990, this molecule used to be considered as very toxic molecule. Even now, also it is a toxic molecule. But in 1998, you can see the discovery relevant to nitric oxide got awarded with a Nobel Prize from the Physiology and Medicine section for their discovery in relevant to the mammalian biology. And what you see is that this discovery was related to the signaling pathway of this particular molecule in the cardiovascular system of mammals. Okay, so now in this case, what happens? Again, this has something to do with oxygen carry, oxygen transport, and so on. So as I said, that this nitric oxide is a molecule which helps vasodilation. So right. So what does vasodilation mean? So that means if you have a normal blood vessel, let us consider it is appearing like this. Now after vasodilation, what happens? The radius of or diameter of this particular blood vessel will get widened. So as a result, what happens? So you would have increased transport of oxygen. As a result, more amount of nutrients will get equally distributed throughout. And then you will have a lower blood pressure. You will have more efficient removal of the cellular waste product. So that means this vasodilation is very important, and then nitric oxide helps in vasodilation and indirectly helps oxygen to flow, or more amount of oxygenated blood to flow. And in fact, nitric oxide also helps or increases the oxygen binding capacity of hemoglobin and myoglobin, right? So that's why it indirectly helps the oxygen binding. So moreover, you can see this is the very recent paper which came out, of course, over the 1.5 years after COVID came in the market, right? So this nitric oxide has also been considered as one of the potential or one of the molecule which which hinders the replication of the COVID virus. And again, this is again in preliminary stage, probably we cannot talk more in details. But again, this is a molecule where people think about. There are significant amount of opportunities there. And then again, as I said, today I do not have time to go into my research. So in our case, what we do is that we try to understand how these different biological pathways generate nitric oxide and H2S, right? And H2S is also another molecule. Today we are not going to discuss it, but that is how our research is connected to these different biologically relevant signaling pathways, right? And then these are the representative publication. Again, this might be too technical for the school level students, but again, this is something which can be handled in a chemistry laboratory, right? But the goal is to understand some of the biological processes at the end, as I was trying to discuss. Do I have what is the time? I think I am overshooting, huh?
Yeah, maybe another 2-3 minutes, no problem. Yeah.
Okay. So I will take just two brief slides just to illustrate some of the other fundamental aspects of nitrate. And so what you can see over here is how the nitrate comes into the body, and then what is the importance of nitrate in the body. So nitrate is also another molecule which is connected to the nitric oxide biology. And again, it comes from different fertilizers. I think all of us know that we use different ammonium-based fertilizers, ammonium sulfate, ammonium phosphate, and then ammonium nitrate. All these different fertilizers we use. Now, in case of these fertilizers, whenever they go into the soil, definitely some part of this nitrogen content will get utilized by the plants to incorporate those nitrogen content into amino acids. That is how the foods are getting generated, right? But a significant portion of nitrate gets just accumulated in the soil, right? So now those nitrates basically also comes into the different kinds of food, including the green vegetables, right? And then also red meats, for example, cow and goat and all other animals will also eat those vegetables. As a result, this nitrate comes into their body as well. So now whenever we consume either vegetarian or non-vegetarian food, so it comes indirectly to our body, this nitrate molecule. And then what happens, you can see that this nitrate is distributed throughout the different mammalian species, starting from human to mouse. And the concentration is quite significant, as you can see over here, although the error bar is also quite high because the reliable method to determine the concentrations are not quite in place, right? But then again, these are in micromolar concentration, which is quite significant for the biological purposes. Now, at the same time, what you can see, the localization of this nitrate in the body is also quite diverse. So that means it is not only located in the stomach, that you consume the food and then it went to the stomach and that's all, and then it will be getting out of the body. It's not the case. It is actually getting distributed throughout the body, including the blood plasma, then brain, heart, liver, and so on. So that means whenever certain species is getting distributed throughout the entire body, that rather indicates that is needed in the entire body, right? So now the question is that how does nitrate gets involved in the mammalian biology, and this is the reaction which is getting actually done in the body. So different metalloenzymes, and even there are a couple of pathways where you do not need metal. It can happen without metal also, but it gets converted. So nitrate gets converted to nitric oxide, and then as a result, it does the signaling activities as I demonstrated in the last slide. So this is the last slide I will show over here. Is that perhaps I think our students should know that Professor P.C. Ray, who worked on this kind of nitrate molecule in the early days of Indian research, and then you can see that this work got recognized by even Nature magazine, and that was the early days of nitrate. But again, in modern day context, what you can see, this is the image which is taken from this review articles. this nitrite is getting involved in different disease, right? For example, starting from stroke to mitochondrial myocardial infarction, then liver and lungs transplantation, high blood pressure, vasodilation, pulmonary disease, and then there are many other diseases which are not included, right? So, but again, all these different routes or all these different possibilities are connected to the various tension metal mediated nitrite to nitric oxide transformation, and as a result, so they do show different biological activities. So now with this, I think I would finish. But what I wanted to mean with this particular overview of this small molecules and the relevant molecules is that these are very small molecules, but again, they have huge impact in case of human or mammalian biology or physiology. And in this particular cases, what you can see is that the tension metals are the one who tunes or they are the one which tunes the reactivity of the metal sites or reactivity of the small molecules. As a result, the different activities one can imagine. So with this, I would end, and I would thank all the participants for their time, and I would then go for a couple of questions. I was seeing there are a couple of questions. Then we go.
Yeah, thank you very much, Dr. Subrata. Can you see those questions?
Yeah, I can see the questions.
Okay, okay.
So the first question is that is there any difference between molecules and particles? Yeah, in broader sense, I should say the particles are just like aggregates of molecules, of course, but again, molecules are like more, I would say, more well defined in the sense that it is just like a single molecule. But in case of particle, it can be a spool of molecules which forms the particle, and then those molecules are basically glued together to make the particle. And again, that glue can be something like the non-covalent interaction. So that means there is some interaction which is not like a covalent bond, but still there is some kind of interaction which basically brings the molecule together and then leads to the formation of the colony of molecules, which leads to the particle formation. Yeah. So now how we differentiate the living particle versus non-living particle? Can we apply physics theory or bio? Like the domain, is it possible living particle with dual nature? Yeah, so I think based on wave particle duality, I think it is possible, but I am not quite sure that how we can directly relate it. But of course, it is possible, and then you can of course apply the physical theory, and of course there are a lot of or significant amount of research which are going on from the point of view of biophysical chemistry or biophysics, right? And again, they also try to understand. For example, at the beginning, I was trying to demonstrate that how does the active site of hemoglobin versus myoglobin leads to the difference in the binding capacity or binding affinity of those particular metal acids. And then those are the things which has been actually deduced by the biophysical chemist. Okay, so that's why and again, there are modelings, and then for those modeling you need those kind of analysis where you will be considering this quantum mechanical calculation and so on, and for that you need those kind of wave particle duality consideration. How do we decide the structure of biomolecule and chemical molecule? Is any device to show the structure of molecule? Yeah, so typically there are various ways to realize the structure of the biological molecule. Again, the question is that how do we realize the biomolecules versus chemical molecules? At the end, it is molecule, right? So everything is made, as chemist, I believe that everything is made of chemicals, right? So now, whenever definitely the biomolecules are the bigger ones, and chemical molecules also can be big, but again, typically we consider the smaller variants. But again, they can be the structure can be obtained from the X-ray crystallography. As I said, that for small molecules, we call it small molecule crystallography or small molecule X-ray diffraction analysis. And then for the bigger molecules, so what one can do is that one can do the protein crystallography, right? But again, the basic fundamental principle is same for determining the molecular structure. And then likewise, if you want to do some other kind of apart from seeing the molecule, you can definitely also look at other property of the molecule or the biomolecule. And again, the treatment is same. For example, if you want to know the strength of a bond in a small molecule, then you can do IR spectroscopy, infrared spectroscopy, to know the vibrational stretching frequency and so on. Then similarly, if you take a biomolecule and then in a given biomolecule, you are particularly interested to understand a bond, you can also do the infrared or even Raman spectroscopy, which is a little bit different than infrared spectroscopy in terms of operation, but then they give the similar information. So one can do that. But again, the fundamental principle for chemical molecules versus the biomolecules are same. And then yeah, is there any other question? We'd be happy to take.
No, I think that you have taken up all the possible questions which are there. And if there is any particular suggestion you would like to give to the students, then I'll be more than happy.
Yeah, so from my point of view, what I would like to give to the students is that many times they feel that chemistry, there is a boundary between, let's say, chemistry and physics and biology, or whatever you consider, right? So what I would say, or with the talk also I wanted to mean, is that actually there exists no boundary. And in fact, not only within science, in fact, there is no boundary between chemistry or physics or biology or even sociology or humanity. So that's why what I suggest to the student is that they should be quite open in terms of thinking, in terms of merging things together, and then in terms of combining things together from the various aspects, right? So that is what I think very important nowadays. So once you know the fundamentals of, let's say, chemistry or biology or physics, right? So now with that given concept, whether it can be applied for another purpose, right? For example, whether a chemical concept can be applicable to biology, or biological concept can be applied to chemistry, right? So that interdisciplinary approach is something that students should think about. And then nowadays world, and you can think about research, you can think about industry, you can think about any other aspect where the science is involved, or even any other aspect of learning is involved. One needs to be multidisciplinary, one needs to be interdisciplinary, right? That is what I think I would say students to follow, and they should not ignore a particular subject and then put all the baskets, all the eggs in the same basket. There are many students who pay attention only to a specific subject, and then they want to proceed with that. That is something I think those days are gone, right? So I think that is the message I would like to give.
Sure. Thank you very much, Dr. Subrata, for enlightening all the attendees, the students, the teachers, and all others who have been present all throughout this webinar. And on behalf of the National Academy of Sciences India Delhi chapter and the Indian Upadhyay College, I would like to thank you once again, and to all the attendees who have been there with us. Thank you very much.
Yeah, thank you, Dr. Samaj. Yeah.